Hello and welcome to this EpigenomicsNet interview today with Ben Laufer, PhD candidate in the laboratory of Shiva Singh at Western University in Ontario, Canada. As part of our focus on pregnancy and the epigenome, Ben's kindly joined us today to talk about his epigenetic research into fetal alcohol spectrum disorders and the potential for epigenome editing as a future treatment for these disorders. Ben will also discuss the long-term effects of neurodevelopmental exposures in general and offers his opinion on where the field of epigenetics may be heading. Ben, thank you very much for joining us today.
Thank you for having me.
So the first question, your work on fetal alcohol spectrum disorders has demonstrated quite widespread epigenetic changes due to alcohol exposure during fetal development. Can you tell us about your most recent studies and their most important findings and implications?
Yeah, so I'll tell you a little bit about my thesis work. So I started on a mouse model of prenatal alcohol exposure. And the model that I used was actually kind of a lower level of exposure than a binge injection. So previously members of my lab had started off and they were examining the long-term effects of a binge injection throughout the different trimesters of pregnancy. My model is kind of continuous throughout all of pregnancy. And rather than, relying on an injection, we actually give the mother the free choice to drink. And the strain of mouse we use actually, chooses to drink a considerable amount of alcohol. That's equivalent to, a pregnant mother who would drink a moderate amount. So even though it's higher in the mouse, their metabolism is a bit different and it proceeds at a higher rate. So in this model, we were very interested in looking at the long-term effects. It's well known that there's, a lot of immediate damage to the developing brain that occurs upon prenatal alcohol exposure. But one question remained is, does any of this alteration remain in the long term, and specifically at the epigenetic level? There was a lot of evidence that, some of the immediate deregulation was caused by alterations to the epigenome. However, the long-term effects, specifically until postnatal day 70, which is the equivalent of, young adulthood in a mouse, remained underexplored. So we sought out to characterize the whole brains of these mice in adulthood, and we looked at DNA methylation, non-coding RNA, and gene expression. And what we found is that there's genome-wide alterations overall. There's no one or two genes or just one pathway overall. It's really this large scale of disruption. But at the same time, this disruption is really non-random. So even though, there may be different genes between the different exposure paradigms that we examined, they usually fall into the same pathways and functional ontologies. So these genes really seem to be doing the same thing, even though there's all this heterogeneity throughout. So one of the big findings from this mouse model was that, these alterations are indeed occurring in the long term. So something may be maintaining these alterations for a very long time, which has given us some insight into any potential for correction. What we also found is that, regardless of the timing of the prenatal alcohol exposure, so trimester 1, trimester 2, and trimester 3, or even the dose, whether it was this binge or moderate exposure, all these different exposures produce alterations. So one big take-home message there is really that, for a pregnant mother, there's really no safe time and no safe amount of alcohol consumption throughout pregnancy. And that was one of the take-home findings there. So after that, we switched gears over to humans. I started a collaboration with Dr. Joaquin Capolanga, who I had the honor of meeting, and he's a MD PhD. And he's working in the clinic right now, and he has access to all these children with fetal alcohol spectrum disorders. So what we did is we proceeded to collect some buccal epithelial swabs from them. And this might seem like a bit of a weird connection, jumping from the brain to the buccal epithelium. But there is this nice little stem cell connection going on there where they have a very, similar shared progenitor, and they're connected at the germ layer of the ectoderm overall. So we seem to kind of, rationalize that if this is occurring early in embryonic development, any stem cells that are affected might maintain some signature as they begin to differentiate into tissues as diverse as the brain and also the buccal epithelium. And the big advantage of the buccal epithelium is we could obtain it non-invasively from these children. It's just a simple swab you take of the child, and we can then assess for any differential methylation. So we use the 450K array to check for any genome-wide alterations, the DNA methylation in young children. And what we found was somewhat similar to the mice. Not identical, but there were still the kind of similarities that we would see if we were to look at the different exposure paradigms or even really, the different amounts overall. So with these differences in mind, what we set out to do is really examine where they laid and what, gene networks, were there any specific loci. And we found very similar to the mice that, these alterations, again, are non-random. We're seeing enrichments for specific processes. In this case, in the children, what we've seen is that there's a lot of synaptic signaling pathways being enriched for. So this is the type of signaling that your brain uses, and it's exclusive to the brain overall. So to see such an enrichment in the saliva of young children, specifically, differential methylation that's related to fetal alcohol, was a very interesting observation. And so these pathways, as I said, were related to stem cell signaling, neurodevelopment, synaptic signaling. And what we also found, though, is that there was a discrete loci, this really broad pattern that was present in the children. This was increased methylation at this locus known as the clustered protocadherents. And they're quite interesting because they generate individual neuronal diversity in the brain. So they're used exclusively in the brain, and essentially they give each neuron a barcode to say, I am me and I'm not you. So when it comes time to form the connections, it takes this into consideration. So really, this whole complex locus we found is involved in just generating this diversity in the brain, and yet it shows a repressive signature in prenatal alcohol-exposed children that we see. So that was one thing that we characterized. We also did a collaboration with EpigenomcsNet where we did some pyrosequencing to confirm these changes, and we were able to confirm them in some of our target genes as well. So overall, it really seems like there's this long-term profile associated with alterations to DNA methylation in fetal alcohol spectrum disorders.
Okay, very interesting. So you're also quite interested at the moment in epigenome editing. We had a focus on that a few months back. Do you think there's much hope for the reversal of these epigenetic changes as a treatment for fetal alcohol spectrum disorders?
Yeah, I definitely see some hope there. And I think there's really two avenues we can use epigenetic editing to study fetal alcohol spectrum disorders and probably other related neurodevelopmental disorders. So the first thing we can do is really, attempt to recreate these marks. Now that I've found these associated marks, we really want to go and show causation. So one thing would be to take a mouse model without any prenatal alcohol exposure and then try to recreate these marks in that mouse and see if any of the phenotypes line up. And then we could have, some causative loci. Then after that, I think the clinical potential will come, because now that we've designed an epigenome editing system to target these specific regions, and we know the effect of, let's say increasing methylation, I would say let's go in and now try the opposite in a mouse model of prenatal alcohol exposure. And theoretically, it should be able to reverse some of these long-term deficits we're seeing. So some of that maintained signature that we saw, I think we can begin to adjust and correct. Now, it's not necessarily going to be a cure, but I think there's plenty of room for improvement overall.
Okay, there's also been a lot of research going on into the long-term effects of neurodevelopmental exposures in general. Which avenues of research do you find the most interesting or exciting at the moment?
I think I'm very captivated by transgenerational epigenetic inheritance. So in my research, we've seen that these alterations are maintained in the long term. It's not just an immediate disruption. Something seems to, be destroying these vulnerable cell types, but then there also seems to be some that remain that are altered and maintain these epigenomic signatures. So, of course, throughout development, there's going to be subsequent cell division. So they seem to be surviving cell division overall. So there is some heritable component there to it. It's really being transmitted. So I think transgenerational epigenetics is really interesting because, we're not just considering mitosis anymore. We're now going into meiosis and the transmission across generations. And that's something that, a lot of people are skeptical about. And that's, where I think the real interest is because, fetal alcohol is one of the good examples for this because there is an excellent example of it causing transgenerational inheritance. And there are a few other chemicals and also life experiences like maternal stress that are capable of causing transgenerational epigenetic inheritance. However, the real question lies is, how many of these life experiences are really causing a transgenerational effect? Is this effect, very select to specific environmental experiences and exposures, or is it more broad? And I think that's really going to influence how we need to proceed with the research overall and how we would go about communicating it as well.
Okay. And over the next 5 years, what are your kind of overall research goals, and where do you see the field of epigenetics in general heading?
So my research goals are definitely to stay in this field. I think it's very exciting. We're definitely at a very critical moment where, all this discovery is coming about, and, it's kind of overwhelming. There's just so many possibilities of things that can, alter a pregnancy. We really need to get a good handle of it overall and find out precisely what is happening, what's going on. And again, similar to that transgenerational route, what is really going on with it. So I plan on continuing on, and I'm going to start a postdoc continuing to examine neuroepigenomics and early life experience. Now, I think in terms of the field progressing overall, that there's a whole bunch of exciting avenues, especially if we're talking about in 5 years. We're really seeing this explosion here. So one that we had just talked about was epigenome editing. I think that one is really going to show a lot of growth. We're going to see many functional genomic studies, as well as the beginning of attempting to correct some of these disorders. And hopefully they begin to proceed with somatic epigenome editing as well. Now, there's more in the future than just epigenome editing, though. There's many kind of promising things, and some other things that I'm interested in is this, idea of using multiple omics to really find a signature. So a lot of the time when we look at epigenetic alterations, it can be widespread, and it often doesn't correlate to the transcription going on at the current moment. Sometimes, this needs to be activated. Other times it might just kind of be a footprint. So to really find out what's causing the active disruption, a lot of the time, a good idea is to kind of overlap the epigenetic marks. So look at DNA methylation, maybe multiple histone modifications, as well as the non-coding RNAs. And if they're all kind of targeted to this one locus, it kind of gives you an increased level of confidence. So I think we're going to see more studies pursuing those multiple omics avenues. Another thing that I think is pretty interesting as well is the idea of heterogeneity. Right now, many studies are kind of they're stuck with these kind of broad tissues they're using. They're composed of many cell types. So I think one thing that we're going to see more of a movement towards is sorting by cell type, but not just bioinformatically, but also physically as well. Just because while bioinformatic correction is very good, there are certainly opportunities for improvement too. And then finally, I think not just the cell type correction, but also getting into single cells, particularly in some tissues that really kind of have individual identities. So I think that single cell studies are really going to enable embryology as well as neuroscience, where those few cells can really have very profound differences, and those can really cause very profound phenotypes. And a kind of final one that I'm interested in, I think, is the idea of these preconceptual effects. So it's not just about blaming the mother. There's plenty of things that a father can experience in his lifetime that can also be transmitted. And we're seeing, new studies on sperm and that transmission's intergenerational transmission. And it seems like there's a very interesting and strong role for sperm RNAs in creating this intergenerational effect. And I think we're going to learn more about that throughout the years.
Okay. Well, that's my last question, Ben. Thank you ever so much for joining us today. It's really interesting.
Thank you so much for having me.
And to our viewers, as usual, please do let us know if you've got any feedback on the interviews. We are on Twitter if you're that way inclined. It's at EpigenomicsNet. Thank you very much for joining us today, as usual. And Ben, thank you very much for joining us again. I hope that you'll stay in touch and let us know how your research continues to progress.
Thank you. Will do.
All right. Goodbye.
Bye.